This is a stop motion displaying the cell cycle. Starting with G1, this is the growth and the normal metabolic functions of a cell. This is where the cell will decide whether or not it should divide. This is the first checkpoint in the cell cycle. This first checkpoint for G1 checks the surface area to volume and cell size. The second part of interphase is the S phase, or synthesis, where the DNA is replicated. G2, the final part of interphase, is where the cell undergoes more growth and energy storage to prepare for division. The G2 checkpoint is where it ensures that DNA replication has happened properly. Prophase, the first part of mitosis, is when chromatid and chromatids begin to condense into chromosomes. Two centrioles move towards the opposite end of cells, the nuclear envelope and nucleolus start to break up, and mitotic apparatus forms. Metaphase is the next part of the cell cycle, where spindle fibers attach chromatin and chromatids together, and all chromosomes line up at the center of the cell, or equator, which is also known as the metaphase plate. The metaphase checkpoint ensures that mitosis has happened correctly, meaning that the chromosomes have lined up and attached to the spindle fibers. Anaphase is when the sister chromatids separate along the centromere and each move to the opposite sides or poles of the cell via spindle fibers.
In telophase, animal cells pinch, creating a cleavage furrow, while in plant cells, they form a cell plate. The DNA relaxes back into chromatin, and a new nuclear envelope as well as a nucleolus is formed. In cytokinesis, now that the DNA is copied and in the daughter nuclei, the rest of the cell divides, including the organelles and cytoplasm.